Okay, hello everyone. My name is Indira Rao, and I will be walking you through my research on the correlation or lack thereof between excess iron accumulation and glucose hypometabolism in the Alzheimer's and Parkinson's brain. Now, I know that's a mouthful, but I'll get to each piece of this shortly. So, neurodegenerative diseases are those that affect the brain and cause progressive cell death. My interest in these stem from my grandfather's brothers having Alzheimer's disease and Parkinson's disease. Alzheimer's disease manifests in the form of memory loss, confusion, the inability to learn, loss of language comprehension, increase in aggression and apathy, amongst others. Parkinson's, on the other hand, is more of a motor kind of disorder. So it has symptoms like resting tremor, difficulty performing voluntary and involuntary movements, and eventual memory and cognitive function loss. Now, looking through the list of symptoms, and if you know anyone who's had one of these diseases, or if you know someone who has to take care of someone who has one of these diseases, you know just how devastating it is. So it's very clear that there is a dire need for some kind of treatment for these diseases. However, this need currently remains unmet. But why is that? Let's look at the causes of the disease in more depth to understand this well. There's no real common consensus as to what exactly causes either of these diseases fully yet, and we're understanding it slowly. A lot of people in the scientific community are inclined to believe that it is caused by amyloid beta. Amyloid beta is a chunk of a very necessary protein called amyloid precursor protein. That's the one over here. So with the amyloid precursor protein, when in Alzheimer's disease, it incorrectly cuts off, causing the amyloid beta to be born. With the amyloid beta, it's a sticky little piece, and it attaches to other amyloid betas that chunk off of amyloid precursor proteins and causes the characteristic plaques that are seen in Alzheimer's disease. The thing about amyloid beta is that scientists found drugs that can clear it out of the brain, but it wasn't very helpful in treating the disease itself. So the attention turned towards phosphorylated tau. Tau in its normal form is very necessary. It holds microtubules together, as you can see over here. The blue line is tau. It has these phosphate groups on it normally, but in Alzheimer's disease, there's hyperphosphorylation, where too many phosphate groups, which is just a chemical compound, attach themselves to the tau protein. So what happens is tau leaves its site where it's binding the microtubule and kind of forms these neurofibrillary tangles by attaching to other tau proteins. Microtubules are kind of like the roads in the cell. So they're used for transport and movement of different substances throughout the cell. When tau leaves the microtubule, the road breaks down and the road is no longer functional. So movement within the cell of different substances breaks down and eventually leads to cell death. In Parkinson's disease, we see a similarity that it is likely caused by these proteins as well that are aggregating aberrantly. So alpha-synuclein is a protein that is found in these little pouch-like inclusions called Lewy bodies in the most damaged cells in the Parkinson's disease, in the Parkinson's brain. So that is the cells that are responsible for movement functions. Other than these proteins, which are biomarkers, there are other biomarkers that are common to both of these diseases. One is glucose hypometabolism. Glucose hypometabolism meaning reduced glucose uptake. Now, it could be that there is reduced glucose uptake, which is the primary form of energy that brain cells utilize. So when there is reduced glucose uptake, it is likely that it could be that the cells are unable to get rid of the amyloid beta or the tau. It could also be that the cells are already damaged by the amyloid beta and the tau, the phosphorylated tau, and this causes them to not take up the glucose properly. But there's more reason to believe that glucose hypometabolism happens first, because for glucose to absorb properly into the cell, we need a hormone called insulin. When insulin signaling breaks down, there is no need for an insulin degrading enzyme, because if there's no insulin, there's no need to degrade it. So when the insulin degrading enzyme doesn't produce, we have difficulty clearing out amyloid beta because the insulin degrading enzyme is necessary to clear out amyloid beta. So with this chain, it's probably likely that glucose hypometabolism happens first. Another biomarker that is common to both of these diseases is excess iron accumulation. Iron itself isn't bad for the brain. We need a certain amount of it for the brain to function well. But when there is too much free iron in the brain, it causes oxidative damage. I won't go into the details of what oxidative damage is because of the paucity of time, but you can think of it in a simplistic way as free radicals wreaking havoc on the cells and destroying them. So oxidative damage is bad for the brain. Iron binds to amyloid beta. So this is important to notice because we see amyloid beta in a lot of places that we see iron in the brain. And I'll get to that in a little bit as well in my results, but it's important to take note of this. So coming back to my research question, the correlation or the lack thereof between excess iron accumulation and glucose hypometabolism in the Alzheimer's and Parkinson's brain. This leads us to a couple more questions after understanding the pieces that make it up. Why are we looking at these two biomarkers, and why is the correlation or lack thereof even important? Let's do a comprehensive literature review to understand these parts. So why these biomarkers? To put it simply, the treatments that are currently being tested for these biomarkers are very, very promising. Now, my mentor, Dr. Fry, came up with the method of intranasal delivery of therapeutics to the brain. So with this method, we, in other studies, they have administered insulin to the brain, which helps with helping with the glucose hypometabolism that I just spoke about. And in other studies, they administered intranasally deferoxamine, DFO, which is a compound that helps clear out the excess iron accumulation. And both of these, this one is, so both of these helped in really ameliorating the symptoms that are seen in Alzheimer's disease, which is the memory function. And in Parkinson's disease as well, they were seen to help a lot. And why is their relationship important? Well, in previous literature, there is an implied relationship. For example, in this 2021 paper that came out very recently, they talked about how when there is, when we clear out the iron, the excess iron that accumulates in the brain, there is improved insulin signaling, which is, a theory. And then in the 2018 paper that I've mentioned over here, they suspected that when there is insulin resistance, which is, again, leading to glucose hypometabolism, when there's insulin resistance, there is an increase in iron load in the brain. So with these studies that have already kind of hinted at this relationship, I think it's worth investigating it to find out what really is happening, to see what the correlation is, and if there is the possibility of a causation between the two. The big picture idea, which is the treatment purposes, is that for treating it, we'll know better if, we need to only administer intranasal insulin, and then both glucose hypometabolism and brain iron accumulation will improve, or we can administer intranasal deferoxamine and both of them will improve, or we need to administer both of them in conjunction to have the best effects. So it kind of helps us understand what the best method of treating Alzheimer's disease or Parkinson's disease is. So my method, I had a tiered approach, and I'll get back to that in a little bit. But primarily, I had a lot of qualitative data, and to analyze the trends better, I made it quantitative, and I'll show you how I did that as I talk further. So I used Fdg Pet Scans and QSM MRI scans. Fdg Pet Scans help detect excess glucose in the brain. So Fdg Pet Scans are kind of helping us detect where there are glucose deficit areas and glucose rich areas in the brain to help us identify where there's glucose hypometabolism. And QSM MRI scans help us detect where there is a lot of iron and where there isn't much iron in the brain, helping us detect excess iron accumulations. QSM MRI is not restricted to iron. It's also helpful in detecting calcium and other things, other compounds like that in the brain. But for the purposes of this experiment, we've looked only at the iron. So coming back to what I did over here using these scans, I looked at different research papers that conducted these scans on Alzheimer's patients and Parkinson's patients. And what I did was, so these were separate studies, like separate QSM MRI on Alzheimer's, separate on Parkinson's, and the same for Fdg PET. So with those studies, what I did was I organized them and I looked at the brain regions that were primarily being affected by brain iron accumulation and glucose hypometabolism to see if they could exist independently or if they always exist together. So coming back to this idea of a tiered approach, my best possible resource was finding a paper that had already administered intranasal insulin or intranasal deferoxamine to see if the brain regions were, showing glucose hypometabolism amelioration and brain iron accumulation amelioration together or not. But because of the lack of research in my field, which is the primary limitation of this entire study, which I'll talk about a little more further too, because of the lack of the previous research, this wasn't a possibility. My other option was to look at brain scans, the raw data, to see if brain iron accumulation and glucose hypometabolism were occurring in the same areas. But this too wasn't very practical. And I was finding data for not age and sex matched couples. So it just made more sense to do it, to find the studies that have already discussed which regions they were seeing it in, to make it a little easier to compare and also make it efficient. So here I have examples of my primary data table, which is also included in my research paper. So going down vertically like this, you can see this is glucose hypometabolism in Alzheimer's disease. So I've listed the experimental studies and the review studies, and over here I have the brain regions. Then I marked whether or not they were seeing glucose hypometabolism in this case, or brain iron accumulation in this case, over here in this section. And then I tallied it up to see where they were, which studies were reporting more, were reporting it in which regions. So you can see over here, for example, in the hippocampus, there were 5 studies that reported glucose hypometabolism. In the cingulate, there were 7 studies that reported it in the cingulate. And then over here, there were 5 studies that reported in the hippocampus, 1 in the cingulate, so on and so forth. Before moving forward, I think it's also worth mentioning that because of my sample size of the number of research papers I had being pretty small because of the lack of previous research, I made sure that I was not introducing more bias by making sure that each of these experiments had a sample size that was statistically significant. And I also made sure that the reviews were not reporting from the same source so that I wasn't over-reporting the number of papers that found a biomarker in a certain region. So moving forward, this is the result section of my paper. So what I found in my research is that in the hippocampus alone, there were equal number of papers reporting glucose hypometabolism and brain iron accumulation in the hippocampus. So in these regions, which I'll talk about first, so these regions in the middle over here, we see only have a yellow bar, which means that there were studies that reported that there was brain iron accumulation, but no studies that reported seeing glucose hypometabolism in this region. For the frontal cortex, it's the very opposite. So what is really interesting is that these first regions, which show both of them, are the only regions that are the regions, I should say, that are primarily affected by the other biomarkers we talked about, which is the amyloid beta and the phosphorylated tau. The other ones are also important to a certain degree for memory skills and are also to a certain degree impacted by the other biomarkers as well. But these are the most impacted and also most important for memory and other cognitive functions like that. So why does the hippocampus and all of these regions show both of them? Well, it could be that when there is both glucose hypometabolism and brain iron accumulation, it could be that together they are causing an increase in the Lewy body, not the Lewy bodies, the amyloid beta and the tau over here in Alzheimer's disease. So from this part, we can see that both glucose hypometabolism and brain iron accumulation can be found in different regions and can exist independently of each other. I started these regions over here on this side just to show that these are the regions that are very important for memory and memory functions. In Parkinson's disease, we see a very similar trend that the substantia nigra, the globus pallidus, and the putamen are all very important for movement functions and are primarily affected by Lewy bodies. But what's interesting here is that the substantia nigra, we do not see any papers that reported glucose hypometabolism. But in the other regions, especially the globus pallidus and the putamen, where the Lewy bodies are seen a lot, we see both glucose hypometabolism and brain iron accumulation. Over here as well, there are regions where we're seeing both brain iron accumulation and glucose hypometabolism exist independently. And there are regions where they are occurring together, which seem to be the regions that are important for this disease. It's also important to note that over here that there are less than 10 studies that even did these, that even conducted these Fdg Pet and QSM MRI scans. So it's also important to take note that these are not conclusive and that there is a certain margin of error because of the sample size being pretty small. That doesn't mean the results are less significant though, because they line up very well with the Alzheimer's research, and also they are quite significant because of the number of papers. So yeah. So what can we conclude from this if that's kind of hard to, because of the limitations, what is it that we can really conclude? Well, we can see that there is evidence to show that glucose hypometabolism and excess iron accumulation are not found in the same regions in Alzheimer's and Parkinson's disease, and that they can exist independently. There are multiple exceptions to this, of course, but it is also clear that they can exist independently, which is quite contradictory to previous research. So what does this mean for any of us? What are the implications? Well, going back to the questions that we asked during the literature review, it could be that both of these are caused by external factors, that there are some external factors. Maybe it could be the tau and the alpha-synuclein or the amyloid beta that are causing these in the same regions. Or it could be that these are both present together and causing the amyloid beta and the tau and the Lewy bodies to be present. As with any research, we're left with more questions than answers at the end, and it makes room for more research to be done in this area. On a more big picture level, this research indicates that a treatment which is kind of giving both intranasal insulin and deferoxamine or any other compounds that help with these biomarkers in conjunction is more likely more effective than administering one or the other. Hopefully soon, with these treatments moving forward and with more research being done in this field, we will have a treatment for Alzheimer's disease and Parkinson's disease. So people like my grandfather's brothers or others suffering from them will have a treatment soon. Thank you.
Thank you, Indira. So you've had an opportunity to prepare for the oral defense questions, and I will be asking you one from each of the categories that you were given: the research process, your depth of understanding, and reflection on the inquiry process. So my first question for you is, how did you determine which results generated by your method were the most important in forming your new understanding?
So I had two very important categories. One was significance and the other was relevance. So in terms of significance, if only one paper reported it in a certain region, then it was very likely not considered. But what decided if only one region found the biomarker in a certain region and I still considered it was the relevance of it to the disease. So for example, if I found that only one paper was reporting it, if we go back to my results, in the cerebellum over here. But the cerebellum is pretty important in Alzheimer's disease. And in the Parkinson's disease, we can see that all of these regions, only one paper was reporting it, and even in the cerebellum here too. But these were relevant to the disease. So these were the ones I kept in. But there were other regions like the red nucleus or the dentate gyrus that are not necessarily as relevant to Alzheimer's disease because they don't play a function in memory, or in Parkinson's disease because they don't play a function in motor skills. So because if they aren't relevant or they weren't significant, I made sure I eliminated these data points.
Great, thank you. My next question is, how do your findings provide direction for future research in the field?
So as we can see, there are a lot of questions that my research leaves us with. One is, what really is the causation factor? So with more research, it could be in the field of, actually administering one of these treatments to either an animal model or a human model, and whatever is approved, and administering one of these to patients with Alzheimer's disease and Parkinson's disease, and even other neurodegenerative disorders for that matter, and seeing if there is an actual change in the brain after brain scans, to see whether, there is an amelioration in both brain iron accumulation and glucose hypometabolism. And in this way, we can really confirm if there is a relationship between the two of them, moving forward from correlation going to causation.
Excellent. And then I do have one more question for you. We're out of time, but I would love to have you answer this question. I'm curious to hear, if you were able to revisit the research process, what would you do differently and why?
I think I would be I think more of the research skills areas where I would focus this question on, because I think I lacked a lot of the research skills when I started, and one of them being communication. I think I would be more open to communicating, one with my mentor and two with my teachers. And I think this would save me a lot more. This would have saved me a lot more time. I think a lot of the times I was doing different assignments and I was doing different parts of my research, but I wasn't very sure what exactly I was doing. And when I reached out later, I realized that it was a very simple thing that I could have solved with more communication. So I think I'd have been more open. I would be more open to communication to save more time and be more efficient in my research.
Outstanding. Thank you, Indira. So this is the end of our formal presentation, and I would like to invite the audience to have an opportunity to ask any questions or have any comments at this time, keeping in mind that we do end at 2:45.